I'm Amy Whitaker, an assistant professor at Fox Chase Cancer Center. When cells divide, our genes acquire mutations that lead to initiation progression of cancers. Mutations can arise either on their own or in response to DNA-damaging environmental exposures, including tobacco, UV radiation from the sun, as well as chemicals in our food and water. Because of the threat of mutations, our cells have evolved sophisticated pathways to repair DNA damage before it results in a mutation. One of the primary DNA repair pathways is called base excision repair. First, a DNA glycosylase removes the damaged base. Next, APE1 cuts the DNA backbone to remove the remaining scar, and the repair is completed when a DNA polymerase adds a new nucleotide and a ligase seals the remaining nick. Hopefully, the polymerase inserts a matched nucleotide because it can't proofread its own mistakes. While a postdoc, I led a study characterizing how mistakes made by DNA polymerase beta during repair are proofread by the exonuclease activity of APE1 before they can lead to mutations. Visualizing the way APE1 repairs mismatched DNA bases is challenging due to the size of proteins and the requirement to see them at the atomic level. Consider the size difference between two objects you're more familiar with, a penny and the moon. If an atom was the size of a penny would be the size of a moon. Molecules this small can't be seen using traditional microscopes. Therefore, to visualize DNA repair complexes, we use a technique called X-ray crystallography. This works by first generating a crystal of the repair protein bound to DNA and then shooting it with X-rays. Mathematical analysis of the resulting diffraction pattern results in an electron density map in which we can build a model of how the protein enacts its repair activity on the damaged DNA. The APE1 protein sculpts the DNA to align its active site with the mismatch. Because the hydrogen bonding between the mismatched bases is weaker than matched bases, the DNA frays at the end, and the mismatched base slides into the binding pocket where specific protein side chains facilitate its removal. In my independent lab here at Fox Chase, we are using a combination of X-ray crystallography and other biochemical techniques to study how DNA lesions in gene promoters regulate the transcription of cancer-associated genes. One way this occurs is through stabilization of DNA secondary structures called G-quadruplexes. Either DNA damage and/or the recruitment of repair proteins to the site of damage can change the DNA secondary structure, which in return activates transcription of specific oncogenes and DNA repair genes. This can impact initiation and progression of cancers, as well as the cell's ability to repair the DNA lesion. Our work aims to reveal both how DNA repair occurs in the context of G-quadruplex DNA and how this DNA repair is coupled to the transcriptional regulation of oncogenes. Importantly, with this comes the identification of new drug targets that can potentially be exploited to improve cancer treatment for our patients here at Fox Chase and beyond.